All right. Shall we give it another minute or so, Ming? Or
yeah, let's start introductions. Let's just start introductions to everything. I'll monitor the weight room. But Danny, do you want to introduce yourself?
Sure. Okay. Well, first of all, thanks a lot for stopping by. I think it's like 110 people or so now, so that's definitely exciting because I think there's never been so many people at once using the LCMS or the GNPS dashboard. So that's really cool. So yeah, I'm Daniel. I just recently moved to Tübingen. I'm a new PI now here, and yeah, together with Ming in San Diego, we're gonna showcase the GNPS dashboard to you today. This contains like a little hand-on experience. So there will be like some links provided to you, or there are some links provided to you in the slides. You could have downloaded. There's a link in the in that Google Doc where you also like signed up to the workshop today. So yeah, feel free to download this PDFs, and then you have exactly what you also see in front of you through my shared screen right now. And yeah, to like keep it like very much in a nutshell, what like the GNPS dashboard does is it takes LCMS or GCMS data, and it basically is a web-based viewer for that data, but also a kind of like interactive platform. So if we look at it in a more like complex fashion, then here the dashboard is in the middle, and we have like all like these different data sources that can go into the dashboard. So including all major MAST repositories. And then we also can upload our own data, and that might be like really exciting if you're already like a GNPS user. We can also directly visualize XICs, for example, from molecular networking jobs, and we can launch different analysis from the dashboard, which will make use of like, for example, classic molecular networking or feature-based molecular networking. So yeah, and in theory, it's a little bit more than just like an LCMS viewer, but rather like an interactive like anchor point of the GNPS infrastructure. And yeah, to like show you this in a basically here overview. So we have data, we can go into the dashboard with this data, but then we can also launch analysis such as feature finding or MAST repository scale spectral searches from the dashboard. And yeah, to like go right in, I provided like here an example link for a classic molecular networking job as well later on for a feature-based molecular networking job. And I really just want to show you this really quick. So if you click on this, you will go basically to the results page of a classic molecular networking job. And if you navigated a little bit through the through molecular networking outputs, you may know that you can now like look for a compound specific information. For example, here we have data from E. coli Nissl that we know produces like the natural product Yersinia bactin. So if I would go here to view all clusters with ID, I basically get a list of all network nodes that are available from this molecular networking analysis. And then I could search now here for the library ID Yersinia bactin, and then I get like different hits. So here Yersinia bactin, as well as like some different metal adducts. So it's a ionophore. That's exciting. And then when I click on the network, on this view network, I can have like this in-browser visualization, and I can like basically explore the chemical space here around Yersinia bactin highlighted in blue. So this is all old news, right? So like I assume many of you have used this, but now actually inspecting the raw data traditionally has been like a little bit difficult. So here we typically had to like open these files in an orthogonal software, most likely some vendor specific software you had installed on your computer. And that like entailed like a lot of clicks and quite a little bit of also like expert knowledge. So you had to install that software, you had to know what software to use and so on. So why not just like clicking on it and seeing your data right in the browser? And I think that's the really exciting part of the GNPS dashboard, that's now available. So if you click on this node, so there's this view clustered spectra link, which basically brings you to the raw MS spectra that are contained in this consensus node here. So if I do this, I will see basically here a list with many MS2 spectra. And now there's this view LCMS chromatogram button. And this is really exciting because now let's say we want to pick like the most abundant one. So I just sorted by precursor intensity. And then I just click on this view LCMS trace button. And then you will directly prod to the GNPS dashboard. So it's really easy and it's perfectly integrated into molecular networking viewer. And then what will happen, it will basically create here such a string, which entails the location of the data and then also like the scan. So it will right away make an XIC here through this field here for the mass 482, which is the precursor mass of like the Yersinia Bactin MS2 spectra we just looked at. And then if we scroll down, we will first see here on the left kind of like an overview of our LCMS REM. So this is a 2D heat map where each MS event is like indicated as a blue cross. And then more importantly for our inspection of this particular node from our molecular network, we will see here an XIC of this compound, Yersinia Bactin. And this is a really nice example because we see that actually we have two peaks here that are nicely chromatographically resolved and have the same mass, so 482 Yersinia Bactin. But when we go back to our molecular network here, then we see that there's actually only one node in here. And I think this is a perfect highlight or like a perfect showcase of, yeah, kind of like the caveats with classic molecular networking. So what happened is that most likely both of those peaks are like isomers from like the same compound, and they have very similar MS2 spectra. Hence, they're collapsed here on MS2 level, but we can like perfectly resolve them by chromatography. So here, it's also a good case where actually feature-based molecular networking would be, yeah, like the better choice. So now the good news is, instead of like rerunning this through MZMine, downloading the data, processing it on your computer, then uploading it to GNPS again, the dashboard also entails a quick feature finding tool. And I think for the sake of efficiency, it might be good if you not all click on that right now on your own computer, but if you want to like play around with it later, that would be perfectly fine. So we can here activate feature finding and basically select MZMine. It's my personal favorite feature finding tool, but there are some other options as well. Anyway, so now we have here some very rudimentary.
One thing I will say for context, first of all, Tomasz and Robin, who are the main people behind MZMine 2, are in the audience. So they made a great tool. You should, thank them. This is not to replace MZMine, especially once you want to do many files, but this kind of integrates it so you can get your first look at the data for feature finding. So it's like you just got the data, you're visualizing, and if you want to play with it quickly, just to save a little bit of time. But then kind of when you're doing like bigger scale and more detailed analysis, I think it's better to go into a specific feature, the actual tool itself, instead of our little kind of quick start kind of thing to it, just as an aside.
Yeah, thanks a lot for jumping in here and pointing this out, Ming. Yeah, that's of course like perfectly important. If you have used MZMine, you probably appreciate like all like the possibilities, both on like the visualization as well as on like fine tuning for feature finding setting side. And here we have like only 5 settings, so this is kind of like very like minimalistic way, but I think it's nice because like the barrier of entry is relatively low. And I think for like a first, idea of what your feature finding might do, I think this is a very quick way. So if we would set now here some default settings, for example, it takes a couple of seconds, and then it would actually perform feature finding here on the fly and indicate now in the 2D heat map each feature that's found as a like a green diamond. And now, for example, here I detect like a lot of features, but I could now increase the threshold, and then here click update feature finding interactively. Now it would take again a couple of seconds, and I would kind of like see like the new feature finding results. So now, yeah, this basically works for one file in here, but once you're like happy with that settings for like, yeah, like a very preliminary analysis, what it also allows you is to actually launch now a larger scale feature finding job, which makes use of MZMine, but as a proteosafe workflow. So here you just hit this button, run feature finding at GNPS with the parameters you selected. And then it will basically bring here this like proteosafe workflow, and you can here select MZMine as a as the feature finding tool. And then when you like go here at the fields, you will see you have the same input parameters that correspond to like the setting we selected before. So now, importantly, you can also select more files. So we only did this for one, and there's like one file pre-selected, but now you can go into select input files and actually pull either from your own user space or from a public massive data set more files to actually like process them all together. And then it's only, so if you submit this, you have to like provide your email, and you have also to be logged in. You submit this, and then you will basically get like an output. Just let me log in really quick. So if I would then go here on my jobs, and I did this before, then I could either download the MZMine results, or here is now a direct link. So that would be the second step. I could click Analyze with feature-based molecular networking, and then actually go with those MZMine pre-processing results directly into feature-based molecular networking pipeline. So for like the time's sake, yeah, we're not gonna go completely through that. If you're like interested and want to like learn how to launch feature-based molecular networking job, we did this in the past, and there's a recording from that workshop. So I would refer you to our documentation page where you can like actually watch it on YouTube. But more importantly, for now, we can just like use this example link and then also open the same network actually from the same data from a feature-based molecular networking job. So this would be kind of like the final result. And here, what we basically have now, we can, again, view all spectra with IDs. So that would kind of like bring us to the Yersinia Bactin network. Okay. View the network, and now here, nicely, you can see already there are 2 ID'd compounds. And when you like look at it now, we see that we have both peaks actually resolved, right? So if I want to like inspect now this data from the dashboard from here, so I can go back to our initial status page, and then go to feature quant detail list, which basically provide me like all features. And then I have here this view LCMS chromatogram link. So here, let's see, 2, if I sort it by the mass, yeah, then I can get here my basically features. And then the same way as before, I just click on the link, and then it will directly open the actual XIC here with our mass and the dashboard. And again, I get like the XICs here. So now what is new for feature-based molecular networking? You can now also select actually all raw files in that initial job. So you will see here that I actually get multiple XICs for all samples that were in this job. So now I could do this for here, my other replicate. So that would be N3, and basically, put them like, in different tabs, or I could like also include them as a second USI here in this viewer. Okay, so I think that's how we basically get here. So now, exactly, one maybe last thing I quickly want to showcase here. So this is not in the slides, but I think this is quite useful. So the dashboard, as I showed you for now, only was used for like one file at once. But for example, here, let's see where this list is. So you see that I actually had like multiple samples, right? And that is really like nice with the dashboard. You can also like do quick comparisons between like two groups when you actually use here the GNPS USI1 and USI2. And what you need is basically just those strings. And I, for example, now here can simply copy them and then actually give like provide the names from the MZML files. So in this case, it would be S underscore N1, N2, N3, and S underscore P1, P2, P3. So I could build this up together. Right? If I just copy paste them. Let's just do it for two now. And then first, like I can again just like look at the XICs. So now here in this case, they would have been already like, yeah, sorted. Like each XIC from those 4 files I selected now are actually like shown here. And then we can see very nicely that we have a signal here for SN1 and SN2. That was the wild type that actually contained the Yersinia bactin gene cluster. And then P1 and P2 were actually knockouts, were like the NRPS genes were yeah, like deleted. And here nicely we don't see any signal. And if we scroll down, then you will also see here that the integration of those XICs is then here displayed with this box plots. So we see nicely, okay, like our wild type has like high signal and the other one not. So this is a kind of like perfect example. If you have some other type of like quantitative questions, you can also do that. You can also limit the kind of like integration barriers. So this can be done here, like through the XIC retention time / integration limits. So if I, for example, would be only interested in one peak, then I could simply narrow this down here. All right, and I think with this was yeah, like the very brief overview of how to like access the dashboard and how to launch analysis from the dashboard in terms of like feature-based molecular networking. And now Ming is going to take over and actually show you how you can also visualize publicly available data such as in MASHub in the GNPS dashboard.
Awesome. Thank you, Daniel. So let me share my screen over here. And yeah, that was a kind of a good introduction to kind of the basic things you can do in the GNPS dashboard and kind of how it integrates into the ecosystem. And so we'll show you a little bit more about how it additionally integrates in the ecosystem and also explore some public data. And so as with kind of most tools in GNPS, or most tools in general, it's very flexible, and we have some ideas on how it can be used. We're mostly wrong, so I will tell you that. So if there's a new creative application on how or where you want to look at data, you know, again, let us know, and we're always amazed by the creativity of the community. But one application that Danny highlighted is the ability to look at your own data, right? As you're doing your analysis, just being able to quickly hop over to the reasonably raw data, look at the chromatograms, look at the heat maps, look at the MS2 that you're hopping around on. We think that saves time, especially this is for grad students and postdocs that look at the data. But then there's this aspect kind of going up the the food chain where whenever you're getting ready as a PI for publication, you want to check the data, right? Right now it might be a little bit cumbersome, but with the dashboard, hopefully it makes it easier to go all the way from final result all the way down to the specific peaks that are of interest. So kind of sharing within the lab. And then finally, post-publication, what I'll show you here, is we want to kind of change this culture about when you do reviews or when you kind of evaluate the data of part of a manuscript, that we make it really easy to start checking results. That, okay, the hypothesis posited by the particular manuscript matches the underlying data. And then, so if that's important for your future work, you can at least start from a solid foundation. And this, as an illustrative example, kind of introduces some of the infrastructure pieces as well. But what we're going to talk about here, it's kind of a little bit natural products heavy, but the same kind of thing applies to kind of metabolomics, traditional metabolomics data sets as well. So this is a paper titled Iron-Mediated Fungal Starvation. I'm going to stop there.
Hi, sorry, Ming.
Yes.
Hi, there's a very interesting question in the chat. It's not from me, but I think it's important
let me pull it up
to answer. Yeah.
Thank you.
Is there a limit to the number of files added for comparison?
That is a good question. So yes, generally we limit it to 100. That's kind of our scale right now. One thing, just kind of giving you an idea of a little bit of the infrastructure. So we can source data from many different locations. So we're talking about public sources like Metabolomics Workbench, Metabolights. And actually, Ewan is one of the developers for Metabolomics Workbench is in the audience, and he was awesome and helped us get the support in there. And then we also have support with Metabolights. If you're on the proteomics side, most of the proteome exchange repositories are supported, and also from GNPS and MAST, so public data sets and private analysis files that you ran GNPS jobs on, as well as you can upload your own. But generally what we do is when we do make this analysis and kind of this visualization, we pull all the files locally. And so we have a reasonably fast internet connection, but it's not infinitely fast. So our general cutoff is about 100 files for visualization. Beyond that, a lot of our visualization throws so much stuff into your browser that browsers generally start crashing. So that's kind of one of the things that we're contending with. As well as if you do XIC on 100 files, we have a reasonable beefy server, but it still chugs a little bit. So that's one thing. And then there's another question: can I export the data in a graphical plot table? Yes. So a lot of the data, there are these. If it shows tabular data, like for the feature finding as well as the integration, those you can export. There's a little export button. For the XICs themselves, we don't have an export button. If that's preferable, just let us know. So we figured the visualization is a reasonable starting point. But we do export nearly all the graphics in either PNG, so if you wanted to paste it in like a PowerPoint, or if you wanted an SVG for kind of Illustrator purposes. So those are all exportable right now. Anyway, great questions, and thank you for bringing that up. It's a little hidden when you're sharing your screen. Any other questions? You can chime in on the chat, or
yeah, I mean, Thomas has another one. Is like there is another one from Thomas.
Cache the raw files. Yes, so preload and cache the raw files. Yes, so basically this is a great, and Tomasz has a history of developing software, so he thinks in this way. And so yeah, what we do is the very first time you load a file, we have to get the file, and so we cache not only the MZML file or MZXML file or .raw file, so we grab that file, but we also reconvert it just in case we need to. So that very initial load for the very first time you look at a file, it might take, depending on your file, anywhere from a few seconds to a few minutes. So this really comes into play during like these large proteomics, like 2-hour run, 100,000 MS2 scan kind of thing. So that might take a few minutes to convert. But then once it's loaded, navigating around is going to be much, much faster. So in the order of a few seconds for even these several gigabyte files. And not only do we cache it, but we also We do some extra optimizations. There's kind of an internal data structure that we load or we create so we can load these files much faster than kind of the original raw MZML file. So it's not great, but it works. It's kind of my opinion on it. Any other questions? Okay, so let's get moving here. And so anyway, broadly, this particular manuscript, I'm not an expert on the biology of it, but this was from one of our friends and collaborators, Scott Jarmusch. And so he's got a kind of a natural products background. And they were just looking at production of particular metabolites in monocultures versus co-cultures. That's kind of the broad outline. But what they did is they actually made all their data available. And when the paper came out, we were able to look at the raw data with all the metadata, which is a kind of a big point that I'll just emphasize. You should always, when you release your data, having the metadata helps the rest of the community. But we were able to find the right data set or the right files corresponding to the right bacteria. And we were able to show what they had in their figures against the raw data. So it's kind of a validation that everything kind of up and down the chain matches. And so what I'm going to walk you through right now is, given that you see a new paper, you see that they've included an accession number for that particular data set, how do we go and start exploring that and validating that? At least one thing that we've done is over the past, we created this maybe 6-8 months ago. And once it was like minimally functional, we were just like going out, new papers came out, and we were like looking and see, hey, we can reproduce this. This is great. Just as a summary, in this particular publication, they had a table in the main manuscript that said, look, we found these new natural products. And they happen to be, a lot of these are variants of existing ones, but these were the new ones that they were discovering in this manuscript. So one of the most basic, it's not comprehensive by any means, but one of the most basic operations that we want to do is make sure, hey, these particular claims of these new metabolites or these new natural products, they actually exist in the data where the authors claim they exist. Not a particularly high bar, but at least from our experience, it's rarely done. This kind of checking is rarely done. So we just wanted to make it easy and go through this process. And so this particular data set has this particular accession at MASSIve. So MASSIve, for those of you who don't know, is the mass spec data repository at UC San Diego. And so this particular accession that you can see at the top, MSV 40856118. So this is one of the data sets that you can that goes along with this paper. And so just kind of walking you through, if you go to gnps.ucsc.edu, there's a little link as you go down, and you should be able to see browse public data sets. So if you click browse data sets here, it'll take you to a list of data sets. So those are following along. We have catch-up links as well, but just for just letting you do it the first time. So there's a little search box at the top. You can enter this particular accession, hit search, and it'll find this data set called GNPS the Streptomyces data set. And so once you found it, you can click on this little green link here. That'll bring you to the data set details. And on this particular page, what you'll find is there's a little button called View Dataset Files in the browser. And so this will give you, so Danny showed a little earlier this little string that's like the USI. That is a representation, or as a shortened version of being able to find a file within the GNPS and kind of broader mass spec ecosystem. But it's really hard for you to write that. You have no idea how to write that string. So what this allows you to do is it lists all the files per data set. And so you don't have to actually figure that out. So if you click on that link, and again, there's a little catch-up link here, so you can go click that to kind of catch yourself up. You'll have the MSV accession populated, and then you'll have a few things that you'll want to kind of start selecting. So the metadata source, we'll want to take the metadata from MASSIve, just kind of as a thing that we'll need to make sure happens. But what you'll see below that are two tables. And these two tables, they're actually identical right now, but each of them lists out every single mass spec file that we can visualize within that data set. So this includes MZML, MZXML, and .raw, thermal raw files. And so what we can start doing is, because we've taken these files that we have in the data set and we've put it together with the metadata, we can filter for subparts of the data that we want to look at. So we think that's kind of cool. This is the power if you include metadata, both for yourself as well as the rest of the community. And so what we specifically did here is, in this top table, and this is what we kind of broadly term as the first group. Kind of Danny was showing a top versus bottom. So here we're trying to find those groups as well. We're going to search for culture bacteria, or culture bacterial. So the spelling you have to get exactly right. And then we filter for those. There's 3 files that meet that particular criteria. And for this bottom table, similarly, we want to search for culture multi-species. And so what this is allowing us to do is, we're defining one group where all the samples are from monocultures. And then the second group, we're defining all the data from co-cultures. And so now we've done that. We've filtered to 3 files here, and I think 6 files on this bottom. There's this other button at the bottom where it says called Visualize All Filtered 9 files. And so what this does is it takes these 3 files plus these 6 files and allows you to link directly to the GNPS dashboard with these files selected, so you can start doing stuff with it. So again, there's some catch-up links that are in the following slides. And so what this will do now is, as we mentioned before, it'll pre-populate the USIs here for the first group. So these are the monocultures, and these USIs here, these 6 files for the second group. And then again, there's this link that you can click. And in the heat map that you'll see that Danny showed off as well, it will only take the very first file on the heat map on the left side, the very first file of the very first group. So we don't show all the heat maps. It's kind of a mess when we try to do that. But okay, we've got this selected. And now what we can start doing is we can go back to the manuscript, and we ask the question, okay, so one of the molecules that might be interesting that they newly discovered was this this particular molecule, this new molecule. What it is they also list out the M/Z that was detected. So what we can do is, in this top right panel, there's XIC options. And so this is an area where you can start entering options to start visualizing and pulling information out of the files. And so you can start entering this manually. So you can copy and paste it, or you can just enter it manually. Anytime you actually change it, it recomputes things in the background. But the server, that's the server's job. If it does allow extra compute, it's okay. So once we enter this 799.445, what it'll start doing is it will start, it'll create a new XIC plot with all the files across the entire chromatography run. So kind of as Danny alluded to before, you have the ability to additionally limit the chromatographic range. And then there's a particular option for that. Or you can also zoom in manually yourself. It's all kind of interactive. So you'll be able to see this little plot below. And so what we can start doing is, we know that this particular molecule happens to actually occur around 2.5 minutes. So if you enter in the XIC retention time view integration limits, and you enter 2.5, again, the units are minutes. And what it'll do if you just enter a single value is it does plus or minus about 30 seconds, so half a minute on both sides. If you want to be more precise, what you can do is you can set a range yourself. And so we don't show the example here, but what you can do is say, if you want to say 2.4 to 2.6, you can write exactly that, say 2.4-2.6, and the system will understand kind of what that means, and you can be a little bit more precise. And a lot of times we don't need that precision because we're just kind of playing around. But you can set those up yourself. So anyway, so that's that there. And then once we actually set these bounds, we can start seeing the peaks that actually occur in the underlying mass spec data. And so we can see the colors of the lines align to the colors indicated here. And so again, we know which files belong to which group up top, and there's different ways you can actually color these things. We won't show that off specifically right here, but there's certain ways to color it. Again, there's like we're kind of skimming the surface instead of features. At the very end, we'll go over some things that I think are cool, just to at least show you off some things you can do, but they don't fit nicely in kind of exploring this data right at the moment. And so broadly, once we start lining up these ED 103, 104, 105, these are the monocultures, and then ED 24 through ED 38, these are the co-cultures. And so I'll spare you kind of the specific details on the claims that they've made in the paper, but broadly, if you kind of look back at the manuscript text on what they say where this particular molecule is found, we can say it's found in the monocultures as well as the co-cultures, so it's consistent with the manuscript. So there's some biological details we're kind of glossing over. But broadly, we were able to get to this particular molecule and the XIC for this molecule reasonably quickly. So that's kind of the goal that we wanted to have right here. So any questions so far? So this is kind of exploring public data. We're going to be moving on to how we can start sharing results and synchronously visualizing results with other people. Any questions? Don't be shy. Don't be shy. Okay. Anyway,
maybe just reiterate really quick what different repositories like feed into like the dashboard.
Yes, that's a great point. So the very first one, maybe I'll just show it off interactively so that you guys can get an idea of where you can select data. So if we go to the GNPS-explorer.ucsc.edu, then that's the place that you can actually start visualizing all the files as part of a data set. And so by default, we enter a massive accession. But what we have below here too is examples from Metabolights and Metabolomics Workbench. So if you have a Metabolights accession, you can enter them, and it's a public data set. Again, these have to be public. If you enter a Metabolights accession here, it'll pull out this relevant information, the data set information, so you can kind of glean that pretty quickly. As well, it lists out all the files as part of that Metabolights data set. And so you can. There's also on the side here. Let me just zoom in so you can see clearly. There's these little check marks. So in case if you don't want to look at all the files, you want to look at a single file, you can click this check mark, go down and say visualize one file that I use as part of this check mark. And then again, it'll enter this USI automatically for you. And I've actually never loaded this. Maybe I have. And it's trying to pull down some files and visualize it for the first time. But so that's Metabolights. Again, you can do the same thing for Metabolomics Workbench that we can load in here. But more, maybe a little bit more natively, let's go to Metabolomics Workbench. So we worked with the team and they did a fantastic job of integrating it directly into Metabolomics Workbench. So I'm just, I haven't previewed this. So I'm just kind of, so if you find a particular data set that's interesting, and there's a thing that says on this right side, download contains raw data, you can click on that link and you can click this that says view individual raw files. And then within this, if these particular raw files are MZML or MZXML, they've generously added a link that says display in GNPS. And so you can go from one click from Metabolomics Workbench to the GNPS dashboard. And then again, this is kind of pulling down some data and kind of eventually loads. So this is the Metabolights data set that we had loaded. Again, I don't know anything about it. It's just a random data set that we pull down. You can zoom around. There's a bunch of MSMS. But anyway, it supports it from all these major repositories. And if you are interested in proteomics data, that kind of works as well. You can enter a PXD and then and navigate around that. So anyway, great question, Daniel. Any other points that you think would be important to bring up? Okay, so let's go back. Oh, sorry. Let me reshare.
Oh no, all good from my side. I don't know if there's more questions from the audience. Don't be shy. Also, if not, just throw them in the chat.
Awesome, awesome. So, okay, so here's a question. I'm probably visualizing my own private LCMS data deposited in a MASHub, even though I can import files from MASHub. Okay, so yes, so this is a problem. If you make a private MASHub data set, so this is not something where you've done an analysis with a GNPS task or anything. If it's literally a private MASHub data set and you have the MSV accession, those are not visualizable. We've basically, you need to run a molecular networking job, or there's actually a summary workflow that makes it easy to pull it in. This is basically a privacy measure. So that's basically it. You can visualize private data only if you run an analysis on it. And the reason we do that is if you run an analysis on it, that means you have access to that private data. So there's kind of an access control layer. And then we allow you to visualize it. So that's, anyway, that's a thing that we've done. But if you made a private MSV data set, they will not be visualizable. We're thinking about adding some of these features for making it easier to review data sets with like reviewer credentials, but those are still in the works. So hopefully that answers the question. But cool. So now one of the key things that I think for the GNPS dashboard, we try to make things a little easier for public data and nicely integrated with public data sets. But one of the key things that we think is unique about the GNPS dashboard that really can only be done if you have this web interface and this centralized web infrastructure is the ability to share results with others. I think there's a lot of really good kind of software that you can run on your own computers, both vendor as well as open source, that can accomplish what we've shown you in terms of navigating around data to the same extent or better. That's but one of the key things is because it's centralized and because it's web-based, we're able to enable you to interact with the data, your data or public data, customize the visualization that you want, and then easily share it with others. So kind of the classic way of sharing it, to me, is you take a screenshot or you do a screen share with somebody else at the same time. And that's great. But one of the things you miss out on is the ability for the other person to take what you've done and build on that particular visualization or explore it in a different fashion. I don't know about you all, but it's frustrating to me sometimes when you're on a video call and you're like, click here. No, click on this button. That's always been a kind of it's always hard to kind of communicate that effectively, at least virtually. If you're in the same computer, that's a different story. But at least kind of when we're doing this remote collaboration. And so one of the things that we wanted to do with the dashboard is the ability to share exactly what you're seeing with somebody else. And so that's kind of this first class of sharing. So if I pick a file, I pick an XIC, I do some zooming in on the heat map, I set my integration bounds, it's relatively straightforward to share exactly what you're seeing with somebody else. Or you can share with social media. That's something that we've done whenever papers are published. And so that's kind of the very first thing. And this is kind of an overview. The second model of synchronization, and this first one is asynchronous. So I send you a link, you can look at it at your own leisure. And the second point is more of a synchronous model. So if I do something, I change the visualization, I want you or an audience to see the results immediately. And so we call this leader-follower synchronization. And so at least the best application that we found where it's most impactful is in the classroom. So whenever you're teaching undergrads or graduate students how to navigate the data structure of mass spectrometry, specifically untargeted mass spectrometry data for the first time, you want to start with, you want to lead them for the first half of the class. And so you have this audience or this group follow along with your particular analysis, and they're tightly coupled. If I do something, they see exactly what's happening. And so my analogy, at least for those of us familiar, is if you use Google Docs or Office 365, you might do some editing of a document and everybody else is viewing the document, right? So they see all the results immediately. And so where we think this is interesting is if you're, especially for a classroom, is you might get the students started showing them the ropes and you take them half the way there. And then at that point, these followers or your students, they stop following you and then they take that particular visualization and they build on that. And so that's a good way to handhold and then kind of explore on their own. And then this final mode is called full sync. And this is meant for, this is kind of the very classical Google Docs, Office 365, where everybody can collaboratively edit the same document at the same time. And so this will allow you to, everybody, if you make a change, you change the XIC, you add an XIC, you add a file, everybody within that session will see those changes nearly immediately. And so there's no, you don't have to say, oh, let me edit. It's just like everybody edits and then everybody gets the edits. Generally, this is, as we've, on Google Docs at least, you don't want too many people. You have to trust everybody in the group as well. So it gets a little messy once you go above just a kind of a handful of people, or at least changing the visualization. So anyway, any questions about kind of the high level, you know, questions about these kind of synchronization modes?
Maybe showcase quickly how to generate the URL of a particular view.
Yes, exactly. So I think that's this next slide here. So within the dashboard itself, there's, first of all, we apologize, there's like a million different buttons and things you have to kind of read and things tucked into some submenus. But one of the most obvious things that we show here is right below the file selection. And right below where you enter the USIs and where you can upload your own files, there's this blue button, large blue button that says link to these plots. And so what this means is this generates a URL for every specific setting that you've entered in this interface. So, you know, how you zoomed in on the LCMS heat map, all the XIC values, the tolerances, the coloring, the styling, all that stuff that's saved within one giant URL. And so what you can do is you can either right click on this link and say copy link, or you can click on it and then go to the URL bar and copy that. And so you can share that with collaborators. You can put that in a Google Doc. You can put that in a manuscript. Or you put that in a PowerPoint just so, or you put it in your electronic lab notebook to say, okay, here I've detected a particular molecule. This is interesting. Here's the link to the actual data and I can go play with it again. And so that's one of the uses that we think that could be helpful. And so this is a way to generate a link that you can share with others, share with social media, share with yourself for this.
Or share with your PI. That's also really cool because as you're doing analysis, you can just share with your PI.
Yeah, force them to kind of look at data too. I know they miss it. So anyway, we hope it's just an easy way to start visualizing and interactively playing with data without saying click here, click here, click here. So it takes a lot of the friction out of it. Beyond that, so that's the kind of URL sharing. So I'll just walk through a few of the steps here, just so that you're aware of where these pieces are. If you want to do this leader-follower synchronization, what you will want to do is there's a sync options in this kind of turquoise button, and what you'll do is if you click it, it'll bring up a little small in-window pop-up. And then it gives you a bunch of these synchronization options. But the most important thing to start selecting is for you to start a new leader-follower session if you were going to be the leader. So what you'll do is under this synchronization type, you'll select leader. And so then what you'll do is click set synchronization, this little button here. And what you'll also get out is a QR code and a follower URL. And so if you want to have other people following it, like if you're teaching a class, give this URL, and again, this is not super long, this is relatively short. You can give this to your class, or if you're doing a small symposium, or even if you're at ASMS and you want to kind of tax our system, you can give people this QR code and say, hey, look at my data on your phones or tablet that they have handy. So this is a URL you can follow that you can hand out, and then as you change anything in the interface, your followers that are on the same page will see that result. So that's kind of the first thing. And there's a couple other things that you can do for security's sake. You don't want followers to be able to hijack the session, so you can generate these unique random tokens. So that's in the documentation, but we won't go into the details here. And then again, there's one thing I haven't shown here is the ability to do a full sync mode. And so there's another drop down here that says a full sync mode. And so there, what you would do is you would share this collab URL rather than these leader or follower URLs, and share with people that you want to work together. And so anyway, so we won't show that live today just because that's crazy with this many people. But what I want to try out, and we've never done it with this many people. I did last week take all the computers in my house, open up all the web browsers, and make 5 different tabs to see how things would go. I don't have that many computers, but I think this is a different kind of test. So if you all would be so kind, if you do have a chance, either scan this QR code with your phone or your tablet, or if you download the slides, there's a follower link embedded here. And what I'm going to try to do is open up the leader. Bring it back.
I also just posted it in the chat in case you don't have the slides handy also.
Awesome. I will close down these slides so that I can copy this URL. So okay, so we're also slowly monitoring the load. So here we go. Let me start changing a few things, and let me just make sure everything's good. Okay. Okay, was everybody who wanted to able to click on the follower link?
Yes.
Awesome. Okay. So this is the highest load. Okay, we're using like 20 cores on the server right now.
So I would ask the opposite question: is there anybody who wanted to follow that could not follow? All right, that's perfect.
Awesome.
Also, if you just clicked on the first link I posted, Zoom actually cut some of the strings. So I just posted a tiny URL that should work.
Awesome. Thanks, Daniel. So let's reload it. Okay, so here, what you'll all see, this is my my leaderboard or my leader screen. So what you hopefully will see on your Zoom.
Ming, one person said using a separate laptop, so 1 second.
Okay, no worries.
One person said their browser is crashing.
Okay, so I will say these work best on like a Chromium-based computer or a browser or Firefox. If you have kind of an older Internet Explorer, which is not particularly supported right now, this is a very particular heavy web application. So they don't seem to handle it very well. So what you might be seeing is like a big frowny face on either the whole website or on certain parts of the interface. Oh yes, so that's a good point. It does work on, so the funnest part actually for when we were developing this between me and Daniel is he would link me to some new data he acquired, and I'd be, we're in the pandemic, so I wasn't actually out, but I was laying in bed, and I was able to click the link that he shared on my phone and just make at least some observations directly on the phone. I thought that was really cool. If you're in a boring meeting and you ever want to look at data, anyway, that's a side point. But was everybody able to get on the website, barring any crashing issues? Awesome. Okay, so what you should be able to see on my screen, I've loaded a new file, and so you should be able to see a different heat map or the same heat map as I'm seeing on the left side. And so what I'm going to do is I'm going to start zooming in here, and what it'll do is it'll sharpen, and similarly, oh boy, let's look at the server. It should hopefully sharpen, and then all of you should be able to. It'll zoom in automatically for you.
It works.
Okay, there I'm seeing the server and it's like really chugging, but it did it. And then what I'm also going to do is one of the things that you can start doing is if you click on these blue X's, and I'll do it. So if you do something, I'll just overwrite your changes. It will show you the MSMS in this bottom plot here, as well as automatically compute the extracted ion chromatogram for the precursor MZ of that MSMS. And so what we'll see here is this XIC showing up on this right side. And so hopefully most of you will have seen that change. And then additionally, what we can see is when we zoom in, there's a little small X here. And what that signifies, this red X, that's the actual MS2 acquisition, or all the MS2 acquisitions across this entire chromatogram for that M over Z. So I think we're reasonably successful, and that worked. And again, as Danny might have shown, I'm actually going to change up here. We have a set of, actually, it's not. We have a preset. This actually happens to be a set of QC standards that Daniel ran. And so what I'm going to do is I have the string here of a bunch of M over Z's that I know occurs in the sample that are the masses of the standards. And so what it'll do is I've updated it, and now you should see the changes where you'll see 6 separate XIC values. Hopefully. Okay, here we go. And so we'll, on my screen, and hopefully yours will be populated soon. I think some of the settings, it might be a little bit different. It might be condensed because that's a setting that we don't save. What we can see is separate plots for, this happens to be a 271 M over Z. This happens to be 278, and apologies, it's a little hard to see on the Zoom screen. 279, 285, 311. And another plot that we also show that might be informative is there's a little heat map for each of the XICs. So one of the things is if you have many different files for a given XIC, it might be really hard to see. Let me just show you when it's collapsed group by file. It might be once you have a lot of different curves. Oh, I keep forgetting that there's a lot of people also hooked onto this, so it's not as snappy as when I'm just using it myself. So you'll see a lot of these traces stacked on top of each other. So it's a little difficult to see when a particular, which file doesn't have the standard or which one has a problem. So these heat maps provide a solution to this. Maybe it's not perfect, but a solution to this. So those are some of the things that you can do. So yeah, limit on followers. Yeah, we try not to go over 50 or 100. We're really testing the system right now, so we want to see how far we can push it. So that was one of the reasons we wanted to try this out. Eva, did you have a question?
Yes, I do. Hi. First of all, amazing. This is so easy to use. I love it. I just have a question. Sorry, I didn't get how you changed from overlaying chromatograms to from stacked chromatograms to overlaid.
Oh, okay. Yes.
Just
Sorry, I glossed over that. There's a little option here called XIC grouping.
Okay.
And so there's 3 options. either by file. So each plot will have all XICs, all M over Zs per file. So then if you have 2 files, you have 2 plots. There's a by M over Z where if you have 2 XICs, then you'll have 2 plots, and then all the files are stacked onto one. And then by group, this is a coloring scheme. But if you have like files on top and bottom on the USI 1 and USI 2, it'll color them differently. So it depends on your particular application. Does that answer your question?
Yes, it does. And I have another question, if
go for it.
Okay. A lot of questions in general. So when I plot the XIC for some of my data, for example, I can't really see any MS2 fragmentation stars, like the red dots in the peaks, whereas I can see that it has fragmented from the heat map and because I know my data.
Yeah.
Is it normal, or why is that happening, or is there an option?
Yes. So there is so this is kind of implicit. There is whenever you put in one XIC value, then that gives you the option of showing the red X's. If you put 2, we have no idea how to like disambiguate it. So then we don't show the MS2s at all. So if you only have 1, that's the only time it tries. Even if it doesn't show up then one of the things that you can do, maybe like it's an isotope, right? That's off by 1 Dalton. Then one of the things you can do is you increase your XIC tolerance, and then it allows a little bigger tolerance for doing the XIC as well as showing the MS2 as part of this. So, and another point I want to just make about the dashboard itself is, I kind of made it before, is like there are kind of standalone viewers, like external software that do a lot of these things and way more. I will admit, like way more. Kind of our goals here are to make kind of a set of features that allow us to do 80% of what we want to do. Then the last 20%, if you want something very specific, I think that's the role of like vendor software or kind of other open source desktop software. That's kind of our zone of operation. But any other, sorry, just interject that. Any other questions from the audience? Don't be shy. Okay, so one other small things that might be impactful for you all is, again, there's rendering options. So you can export PNG or SVG. By default, it's an SVG. And so for each of these plots, there's a little kind of camera icon. So if you click that, it'll save out a file, and this happens to be an SVG. So you can see it's vector. You can bring that into Illustrator or Inkscape or whatever your favorite thing is. There are other options. You can change some of the styles of the plots, just theming in general. If you like perhaps like the ggplot one better, or you like a Seaborn one better, we just kind of stick with this default one for now. And the one thing that we have also done, this is mostly for me, is under visualization options, you can change the heat map color. Again, there was a question about heat map, so we'll get back to that. But you can change the color scheme on the heat map on the left-hand side. So by default, we do this reverse hot, but there's a whole set of options depending on how you want to visualize it. You can also change how the MS2 markers look. You can change the size and you can change the color. So depending on if there's a particular style that you particularly like. And most importantly, there is this dark mode option. So it changes both the plot style as well as the heat map coloring thing. So anyway, I like it a little bit more for certain applications. You can see a little bit more dynamic range. But going back to the heat map, so I think
I think we should highlight the dark mode that is generally not preferred for journals. So I agree with Ming. It's easier to inspect the data and you can get sort of visually a higher dynamic range. but journals in general do not like dark background. So if you were to go to, let's say, a Nature journals or Cell journals type paper, they generally will ask you to convert a black background back to a white background.
Yeah, that's a great point. That's a great point. So again, you can change all of these things about it. There are a couple other just kind of ancillary features in case you ever wanted to compare two files side by side. So you're going to see some stuff happening on your screen. So my apologies. One of the things you can see is the heat map on the left-hand side. You can also have a second file under this USI that will show up as a heat map right next to it. So the server is struggling a little bit, so hopefully that'll show up and you'll see these kind of things slide on down. And so the unique portion, let me also remove the MS2 markers. So one of the unique things is if you want to compare two files and see if it appears in one versus the other on the heat map side, if you zoom in on the left-hand side, it will lock the zoom on the right-hand side. So this is a particular feature that I thought was really neat in OpenMS, the top viewer. So we kind of implemented it here as well. So you can kind of see these things side by side. This is not particularly interesting, but if there is a particular, different kind of conditions, or you change kind of your mass spec acquisition methods, that might be. Again, about the heat map itself, and so what we've shown here is, I think a lot of people traditionally probably look at TICs and look at it that way. This is the influence of this comes from proteomics. So my background historically was proteomics, and this is how I always viewed the data when I was a PhD student. And so on the x-axis is the retention time, the y-axis is the M over Z, and then the heat map itself, the color that you impart here is the intensity of the MS1 information. So you can start seeing, if you start zooming in here, let me also change some of the quantization so it's a little bit clearer. And so it kind of helps you look at this two-dimensional feature bump, right? So in LC, it comes out in retention time, so it becomes less intense, more intense, less intense. And then you also kind of see the isotopic distribution as well. And so you can see that in the M over Z, where the different isotopes, depending on the composition of your particular molecule, it can decrease or change in relative abundance of the isotopes. So that I thought was kind of a cool way to look at it. And that's very natural to me. I think Danny didn't like this initially, but he's come around to the idea. And so anyway, that's one of the things that you can do. Additionally, let me just show you another item that I found was informative. And this comes from Catherine Maloney, and they've been teaching using this at Point Loma University for undergraduate chemistry students. They just actually did a session last week. And these are classes relatively small, about 15 to 20 students. But what I thought was kind of cool about using the dashboard is they were able to have students explore data. And these are undergrads, and they did it in a linguistics lab. So linguistics computer lab. So it kind of shows the portability of it all. But one of her suggestions is kind of a more natural, I think, coming from vendor software, is if you click here on the TIC plot, we find the closest MS1 scan and show you the MS1 scan. So this is kind of ergonomics for people that are more familiar with that kind of feature on it. And then let me think of any other small things that might be interesting. It also remembers the history of your analysis in this particular session. So if you look at your data and you go through some steps, so imagine you zoomed in, you zoomed out, you added an XIC, you added other XICs within your particular session itself. It actually remembers each of the steps. So I think, well, anyway, I think maybe we're syncing, so it doesn't remember. So anyway, there's a feature for that, and you can replay in advance as well as share those with others. So anyway, that's kind of a feature tucked in there that we won't go over too much. And finally, there are, here's one thing that I'm personally just curious. So we're going to try loading an enormous file for proteomics data. So
before you go like to the big file, do you want to show quickly how to launch like a MAST search?
Oh yes, yes. Oh yeah, that's a great point, Danny. So let me select.
Is there maybe we should first ask the question: do people know what MAST is, or is there anybody that does not know what MAST is?
Well, let's recap really quickly. It never hurts to recap.
Yeah, no, that's good. There are people saying that they don't know what MAST is, and so it may be nice to highlight. It's one of the most amazing tools, I think.
Well, I don't want to toot our own horn. I think it's okay.
To me, it has a big impact. I use it all the time myself, so it's really quite nice.
First of all, yeah. So the way we think about MAST is, so you have an MSMS spectrum in your sample, right? And that's cool. That's interesting. There's many different things you can do with MSMS data. The first is, let's try to match it against spectral libraries to figure out the underlying structure, right? So that's kind of the first answer you want to ask. Other things as a chemist and mass spectrometrist, you might be able to divine, oh, this, I see a certain fragmentation that's interesting. It might indicate a certain substructure or some things you know about the molecule. However, in a lot of different cases, you might not be able to figure that out. You might not be able to know the structure just because the libraries, we're doing our best to expand them at GNPS and the community is adding to it, but we're nowhere near identifying all MSMS spectra in the world. So one of the things that you can do, that MAST enables you to do, is to ask a different kind of question. Instead of what is the molecule that generated this MSMS, you ask, where else have I seen this MSMS before, ever in public data, in all public MSMS data? And so what this answer will give you, hopefully, is some sort of context about where, maybe this particular MSMS is unique to a particular type of sample, a particular type of plant, or simply it will tell you that this MSMS is unique. It hasn't been found in any other public data. So from a natural products standpoint, that's somewhat interesting, right? Novelty is cool. In other kind of situations, you might see that, oh, it's found in only skin samples, only stool samples, or only in a particular type of bacteria. And so maybe that gives you some context of like, okay, I don't know the structure, but it gives me some biological information that I can. So
can I highlight some examples of how people may want to use MAST? So let's say you're working in an animal system, and you want to know if there's a chance if that molecule is made by microbes and not really by the host cells. And so when you do a MAST search and you start to get lots of matches of that MSMS spectra to microbes, then you can formulate a pretty good hypothesis that it's likely a microbial molecule. Another example would be, let's say you're studying an animal model system of a disease, and you want to know if this is translational to people. So you find a new feature that is associated with disease, and now you want to say, okay, is that feature also found in humans? You can just simply search that MSMS spectra, and if there are matches to humans, then you at least know that it's going to be translational. Or if you're interested in natural products and you want to look at geographic distribution, that is one way to find out, because if the metadata that is reported is redo based, which also provides geography if that's provided. And so there's lots of places that this can be really nicely used and can help you advance your science quite a bit.
Yeah, one idea that one of my friends, Vanessa Phelan, kind of thought about is if in your sample you see an MSMS and you want to purify it, and it doesn't produce, it's not produced in very high abundance, you can start asking where else has this molecule or this at least MSMS occurred, and that might be produced in a higher abundance, and so I can grow that bug instead of my bug that doesn't like to produce it very much. So I thought that was kind of interesting, at least to me, who's
it's a
never
great
really
application.
But anyway, now that we got that background out of the way, first, if you have an MSMS spectrum in your sample, first of all, you can see the MSMS spectrum here. and that's kind of cool. And as an aside, there's a little button here that says View Vector Metabolomics USI. There's a other interface that we kind of integrated in. If you ever want to make fancy vector-based visualizations for MSMS to put in papers that you can customize, that's a thing you can click. But what you can start doing is, if you click this button called MAST, Spectrum, and GNPS, it'll automatically take you to GNPS, pre-fill out the precursor M over Z and the fragmentation peaks, and then all you have to do is click go. Oh, title. Okay, so you have to put a title. So that's probably good. Test from GNPS dashboard. And then minus that, you can quickly hit go. So that's kind of how a lot of these things nicely integrate to the rest of the ecosystem. And let me think of any other. And also, if you want to see examples, we try to kind of list out all these different examples here, and they're simply URL links, and you can click them yourself for different ways to look at different data, different types of data, different sample types, and different data sources. So that's kind of this big surface area we want to cover. So hopefully that kind of gives you a little bit of context of the things that we imagine you could do with the interface, as well as you can download this particular data set here for if you want to just have it locally. And let me think. Oh, at the very bottom, there's a bunch of debugging things for us, so maybe ignore it. But if you, in the same way, so just in the same way that we have this link to these plots up here, and it gives you a URL, for the time being, as long as the URLs don't get absolutely massive, for more basic things, and we're still kind of struggling with making the URLs a little smaller, is we have a QR code for exactly your analysis. This is like the biggest QR code I've ever seen, and this is pushing against the limits of QR codes. But technically, you can post this in like your, if you have a talk or your ASMS posters and things like that, you can post it here and say, hey, use your phone, bring it up to it, and you can look at my data. Especially like if I know some grad students and postdocs that they're very proud of their data and they're like want everybody to look at it. So this is a way to kind of share it with the rest of the world. So anyway, that's some of the things that we wanted to show off here. There's even additional filters. Oh, one kind of quick idea is if you have polarity switching as part of your data, you can view that data here, but you can also copy and paste the same file in USI1 and USI2, and then you can actually filter for only positive data on the left side and only negative data on the right side. It looks kind of cool if you zoom in on the same area, you see the off-shifted positive and negative mode ionization for the same molecule. I thought it was cool. And there's also other features about overlaying feature finding information onto these visualizations. Kind of you can arbitrarily, we won't talk about that in too much detail here. And also for those of you that do targeted analysis, we have these options in advanced. Up here, there's an advanced XIC options. This doesn't particularly have it, but if you do SRM assays, actually, let me see if I can pull up an SRM example. It won't be part of the leader follower thing. But in this SRM example, these happen to be, we worked with Michael Marty from Arizona, and he was teaching this in his class. So we're like, well, let's do it and see what happens. And so within these particular samples, they have different SRM assays. And so you can select them, and they will natively show up as part of as an XIC plot. So if you do targeted acquisition, you can use this to share your data as well. As well as in his sample, he also collected UV and then other kinds of visual data, I think. And these also can show up as chromatograms. So it's reasonably versatile from that perspective. But anyway, any other questions? We've covered a lot of ground for such a simple tool. Don't be shy. You can ask now. You can stop by GNPS office hours. You can put it in the chat. Okay, so repeat the M over Z versus. Oh, okay, so the M over Z. So this heat map, is that what you're referring to, Katarina? So this depends on your particular application, right? So sometimes you want to look at a particular isotopic distribution in 2 dimensions. So if you look at an MS1, that only is in the MZ dimension. If you look at an XIC, that's only the RT dimension. But sometimes you might want to start zooming in, and as this kind of looks a little bit nicer, you can see the distribution in 2 dimensions. So you can, okay, so the resolution is a little bit low for me, but you can see how this particular feature evolves in time and M over Z at the same time. So in one particular plot. And so you can, one of the things that you can kind of see very easily at least when we did this in proteomics, coelution is a pretty big problem, especially for MSMS. So you can kind of see in these 2 dimensions, if there's coelution occurring between 2 molecules that starts interleaving in retention time and M over Z. So that's one of the ways that I personally looked at it. But again, maybe Daniel can explain it better from a mass spectrometrist perspective. It's okay if he doesn't want to do it.
Sorry, what do you want me to explain?
So how do you use the heat map? How would you explain what is the utility of using the heat map versus
so for me personally, I think I'm most interested in like the placing of like the MS2 events. And I don't know, do you want to like just like toggle the MS2?
Yeah, let me change that so it's a little. Okay,
thanks. So I think that the awesome part of like the heat map is that it gives you like this kind of like bird's eye view of like your data, right? And especially like as our pipeline is like mainly depending on MS2 scans, it's really essential that like the DDA, so data dependent acquisition, and your method does a good job and you cover kind of like the whole ground like a little bit. And yeah, I mean, if you just like zoom out, then I think one can like easily see that there's a lot of like this white crosses in this case with the reversed view, and a certain like area of like this chromatogram where the highest intensity is covered by those events. So I think that is really useful. Otherwise, I think it's kind of like just like a top view of like your TIC. So it's also kind of like resolved by MAST, but sometimes it's like hard to judge what like the abundance differences are based on the heat map. But yeah, I think it's just like a very first navigator. There are other tools. I think like OpenMS has also like a very similar view, and I think also some vendor softwares have like similar views and with a 2D heat map. So yeah.
No, that's a great point. I think one of the things that we see in our data is if you set your dynamic exclusion parameters improperly, it's pretty obvious. In this view, you look at it and you're like, oh, okay. So
so then you would get like this horizontal lines and basically see that like the MS was triggered like for the same ion over and over again. So I think it's a very first check of like data quality as well. So
yeah, and it depends. Yeah, so if you want, I guess there's a comment in the chat. If you want dynamic exclusion, if you don't want that, then that's kind of a person, kind of your choice on your experiment. But it allows you to kind of visually very quickly inspect, okay, I expect a particular pattern of my MS2 acquisitions. If that's not happening, then you can immediately go back to your instrument. And the question is, another question about MSE data. So this interface should have no problem with MSE data. What you will actually see, do I have MSE data as a preset here? I don't think there is MSE data, but there is.
I think the GCMS data is the closest format.
Yes, let me look at sort of, yeah. So there is GCMS that you can view. And the thing that you'll see with MSE that is interesting or somewhat interesting is very similar to swath data. So this happens to be DIA swath data. And so what you'll see, and in difference from what Danny was talking about, like these MS2 points depending upon the data, and it's only where the heat map has actual signal. With swath data, kind of by definition, it's data independent. You see these straight lines, right? You see it, MS2 scans everywhere. And so you'll see and as I zoom in, you'll see, again, these blue X's are MS2 scans for the swath DIA. But for MSE, what you'll see is instead of taking small windows, it takes the entire MZ range of the precursor and fragments it. And so instead of multiple horizontal lines of these blue X's, you will see only a single horizontal line. And I've actually looked at some myself, and it works. Whether you can pull, how you want to interact with the data will be a little bit different relative to kind of its DDA work, DDA data, but it should support it. Any other questions?
Yeah, I had a quick question actually. So I've been kind of following along with you and my own data set on the dashboard. And every time I select like a new file, it kind of comes up, the heat map almost comes up identical to the other ones. So it's like they're kind of like these squiggly lines across my entire heat map vertically, if that makes sense, for every file I open.
You mean on this left-hand side here?
Yeah, on the for the data
Why
exploration.
why don't you share your screen if you're comfortable with it?
I can't because I am using I have to use my laptop to do all the GNPS stuff on, and I'm using my personal laptop for Zoom. So
okay.
I can't share my screen.
I
Sorry.
would recommend you send us a URL, but then it's kind of public, so that's also an issue.
Yeah, I can't do that either. My PI would kill me, but it's okay. Maybe I'll just go to office hours then.
Yeah, hopefully it's a small thing. Or I mean, there's bound to be a million bugs under the hood. So so we'll get it resolved. But any other questions, fundamental or technical, or just suggestions in general?
I have a question again. And
Yes.
I was wondering about the feature finding features that you have in there. So you have a list of quite a lot of options, but do they all work, or is it as MZMind right now the one that you suggest? Are you going to add also new ones like the OpenMS feature finding maybe, or?
So yes, so that's a good question. So as we look down here, let me zoom in just so you can see a little better. So the first two you don't want to use. So it's test and trivial. So those are for me. So just to make sure like kind of basic operations are working. MZMind is the one we recommend for metabolomics data, just because it's the one we use quite commonly. And Dinosaur is another one that we use for proteomics. And kind of the reason that we chose these is because they're fast. It needs to be responsive. There are other ones that are not fast. And so additionally, kind of our thought process here is it allows you to explore your data quickly and optimize some parameters quickly. And so we include specifically MZMind here because you can do it for one file, and then we have a downstream workflow where you can do it for hundreds and thousands of files, right? So that's kind of the scale that we personally operate in. With regards to other feature finding tools, we are working. So one of the things that we have in the works is Robin is here, and he's working diligently on releasing MZMind 3. And so that's another thing that we have a beta version, but we want to hook up everything so we have a downstream workflow for that as well. So that's kind of the support that we offer. With regards to OpenMS or other feature finding tools, if you would love to help out and contribute the software and the templates and what would be the recommended set of parameters that we can change, we would love to kind of integrate that in. One of the things that is missing is we don't have like a downstream component where you can scale that up from one file to more. But so that kind of like it's kind of a buzzkill, right? So you detect one file and you're like, oh, okay, let's do all of our other files, and you're like, no, sorry, that's it. but you know, if that's acceptable in terms of a user experience, you know, and you'd be happy to kind of work with us to incorporate the code, we're happy to do it, as long as it's reasonably computationally efficient. That's kind of just because everybody shares the same resource. So if it takes like 10 minutes to do feature finding, then it doesn't make any sense. That's actually
So I think so certainly if people are willing to provide the code and adapt, so OpenMS could be adapted for it, that would be great. But you have to keep in mind the dashboard is designed for quick inspection of the data. If you want to do deeper analysis, I would use the feature-based molecular networking workflow. I put the paper in the link. And OpenMS is one of the options that you could use with that sort of strategy.
Yeah, exactly. So, but again, it's like, if you want to put in the effort, we're not going to say no, as long as it fits into that ethos of kind of quick inspection, making things easier, and using, it's really a gateway, right? So it's like, we want to make this a gateway to other tools, right? Either within GNPS or outside of GNPS. Like one of them is like to get you started to do feature finding in MZMine, right? So it's like, if you think that it provides value, then you can go do MZMine on your own with more tweaking and things like that. And we are never going to provide the same level of detail in customization as MZMine, but that's not our objective, right? So
maybe to add one info there. So yeah, this is really meant as like kind of like a quick entry, but besides like launching larger MZMine jobs on like the Proteosafe workflow, you can also download the actual batch file, which will provide you kind of like a starting point when you want to like run the GUI on your own computer.
And yeah,
I was able to, sorry. Oh, sorry.
Go ahead.
I was able to get onto the Zoom call from my other laptop. So could I share my screen?
Yeah, let me add you as a co-host. Okay, go ahead.
Okay, great. So this is what keeps coming up for the heat maps, actually.
Oh, perfect. Okay, so here's what's happening. So if you scroll up really quickly, you'll see the USI it has a scan number associated with it, which is exactly what we want. So now we're zoomed in. So if you scroll down back to the heat map, so what you can see is the M/Z range is 313 to 318. The retention time range is 18.8 to 19.6. So we're zoomed in. So what you can start doing is if you move your mouse over the heat map and you go to the very top, there to the middle next to the home button, or you can zoom out. You can zoom out, or you can do the home button. Either one will work. Reset axes. So you click that.
Okay, great.
So when you actually clicked on it, you were looking at a particular file, so it zooms in really deep there. So let you look at the neighborhood. But then generally, in some ways, sometimes people leave off the scan number on the USI or the links we have, leave it off. And so it'll view the whole thing.
So then the same thing's just happening here with this other one. Exactly. I just need to like zoom it out.
Exactly.
Okay, great. And then I had one more question, because I also noticed that I keep getting this spectrum placeholder. What is this supposed to be for? Is this supposed to be like the mass spectrum?
It's for the MS2 or MS1. So if you want to look at an MS1, click on the TIC, anywhere on the TIC plot. So click on, so that'll populate it. So, or if you click on an MS2, it'll show up there. So either the blue X's or the red X's, that's just the area to show an actual MS scan. So.
And I think this is also a great example for like the comment about dynamic exclusion, right? So when you look at like the heat map, so you see in this case, maybe that was wanted from your method development side, but there is seems to be no dynamic exclusion being used. So you get like this vertical lines of MS2 events.
Is that a bad thing? I'm sorry, not that I wasn't paying attention, but I'm really new to all of this. I'm a first-year grad student, so I'm like super lost about all of this.
I think it really
ask as many questions as you want. That's what we're here for.
I'm sorry, what was that, Danielle?
So yeah, I think it really like depends what you want. So if you want to like only cover like the main compounds and get like many MS2 to like pick the highest quality in a kind of like targeted fashion, I think it makes sense to not have dynamic exclusion. If you want to have like a broad coverage and you want to like get as many MS2s of like the entire kind of like chemical space your sample covered, then I probably would use dynamic exclusion.
Okay, and is dynamic exclusion related to data dependent acquisition?
Yeah, exactly. So it's I don't know what platform do you use here. Can you scroll down for a second? We actually can see it on the dashboard as well. All to the bottom. Oh yeah, so this is a LTQ Thermo. Yeah, I think that's definitely there in like the data dependent acquisition settings. You may want to like optimize that, but often it's just like a couple of clicks in your method editor. And what it will do is basically after an MS2 is triggered, it like sets it on an exclusion list for a defined amount of time, and then it doesn't repeatedly like trigger like the same ion over and over again. And yeah, you can like set this basically number of scans till it's like actually put on the exclusion list, and then also like the duration of the dynamic exclusion. So kind of like if you have an isobar that like elutes like a couple of minutes later, you know, you want to like kind of like clear it up periodically. So in case like another molecule with the same mass elutes later, that this gets like considered again for MSMS.
Thank you so much.
Sure. Also, I don't know if you want to like look through it, feel free to stop by at the office hour, preferably on Tuesday. I'm happy to take a look at it.
But yeah, any other questions? Awesome. Well, if you do find bugs or if you like it, that also, you know, we would love to hear from you. And if you're going to use it in a class, just give us a heads up just in general. I don't think we're going to, I mean, just in case we do maintenance on any particular times. But yeah, if you want to do it in a class, feel free to do that. We've done it at UCSD a couple times, at Point Loma, at Arizona. So it seems to work out, and hopefully it helps. At least you don't have to install any software. So I think that's at least a bare minimum of a win. So, and also I'm personally just curious on like if you for the grad students and postdocs out there, if you share with your PI, it's like, hey, here's my data and here's the molecule. Are they intrigued? Do they actually want to look at data, or maybe they just want to look at a finished paper or figure? So this is something I'm curious about.
Is Louis still here on the chat? Because Sydney asked about the bioactivity-based molecular networking method. So basically, can we request for an easier alternative aside from R to compute the correlation p-values for the feature-based molecular networking profile? Basically, correlate bioactivity with features that are observed by mass spec, which is what the hallmark is of bioactivity-based molecular networking. And right now it's just an R script, and we don't have a systematic way of doing it in GNPS yet.
But cool. Any other questions? GNPS or otherwise, or mass spec? Well, we hope that this changes some things within the field about just making data easier, more accessible, and also just encouraging people to analyze and look at public data. So hopefully that, you know, you learned a lot here, and don't be shy about looking at new papers and the data set that was recently published. And as a minimum thing, when you review papers, when I review papers, the thing that I request as a reviewer is I need to see the raw data. There's like no way around it. I refuse to review a paper unless the data is available. And so now hopefully this is another easy way to, given that the data is there, you can start visualizing it and exploring it and confirming it. So you have that power as reviewers. hopefully you decide to use it. So anyway, that's kind of my shtick on open
So
data.
I think this is Savannah made a comment. I work under Dr. Skylar Carlson, and I know she wants to see all the data. This is ideal for that.
Yeah, no, that's great. That's great. I mean, Peter, I don't know. You tell me, Peter, how much raw data have you looked at, in 2019 versus the last 6 months?
How about the last decade?
Sure. Whatever range you want to say.
It's probably 100 to 1,000 times more than I've looked at in the last decade alone. And so it makes it really nice.
So anyway, I've looked at a lot more data too, just kind of for fun. And you just get a better sense for like how different labs and different groups are acquiring their data. And then it gives you ideas. It's like, it makes you think about why fundamentally does mine look different, or what are the commonalities, what are the differences? And it's just a way to learn instead of just talking about, oh, set this setting, instead of looking at the outcome, right? Or at least a overview of the outcome. And then there was one question, is there a difference between the treatment of GNPS with raw files versus converted file? So if you have a raw file from Waters, so this is the question regarding Waters, you can't view it on here unless you convert it to MZML. So you just have to convert to MZML. If you have a raw thermo file, which we can convert natively, then we just centroid it and it comes out to the same thing. I think thermo's converter within MS Convert and ProteoWizard is the same as this other converter that we use, which is the thermo raw parser. So it should all come out to be centroided peaks. You can, I don't think you can look at profile data in this because we force a conversion to centroided as part of our initial load. There's just too many data points whenever it comes to profile mode. So that's kind of a design.
I have a comment. I have a comment on that because I've been working a lot with the centroided data, converting them, and then using OpenMS workflows. And actually, the converter from GNPS doesn't work, so it crashes. The feature finding Metabo from OpenMS cannot read the data, which is really weird. And then, yeah, sorry.
Okay. This is an OpenMS problem.
Okay.
So yeah, so this is like specifically there's OpenMS has if it's newer MZML files, they write specific controlled vocabulary fields into the MZML file. OpenMS sometimes doesn't understand that controlled vocabulary field and crashes.
Amazing. I lost a lot of months of my life with that.
Yeah, so one thing you could think about is convert to MZXML and then use the OpenMS converter from MZXML to MZML, and then you use their tools.
But
maybe that's what you can fix when you go and visit his lab.
Yes, we will talk about it for sure.
Exactly.
But then the MZXML file actually has less masses than less features than the MZML sometimes. So I don't know why from the ProteoWizard conversion I'm getting less ions, which I'm sure
that the case. That really shouldn't be the case because the data
I went really deep into that conversion thing, honestly.
Yeah, that's weird.
But the MZXML was the first initial converter, right? The prototype. I don't know.
Like the older data format, right? So MZML is kind of just like the newer version.
Yeah. So maybe there was this. I don't know. I don't know why.
It should have the same information though.
Yeah, that's really weird and it's really worth bringing up, right? So anyway, yeah.
Do you think it's a ProteoWizard issue? If that's the case, we should reach out to them.
Yeah, I should probably, because then I'm using the GUI and the and the command line, and I'm getting different results. So I should probably talk to them. They haven't updated it also for many years.
Correct.
Yeah.
Because there's no active funding for it.
Anyway,
I'll email them.
if also for those of you who want to contribute, there's not much active funding for GNPS dashboard either. So if you have an idea, we can work together. Okay, well, we're gonna call it. It's close to 2 hours. We said an hour, but there were a lot of really great questions. What we'll do is we will take this video and we will upload to YouTube after a bit of editing. You know, thank you so much for stopping by and being engaged. And let us know what you think. And hopefully it's useful. At least it's been useful for me and Danny to like look at each other's data. He like runs something like an hour off the instrument. He's like, Ming, check what I did today. And I was like, oh, that's cool. So at least it's one way to pat your lab mates on the back at a very minimum. So anyway, thank you all, and stop by office hours. We'll probably do another workshop for kind of more in-depth things about this, but I think we actually covered a lot of ground. But anyway, thank you again, and stay safe out there and take care.
Bye. Thanks for stopping by.